So thanks a lot for the opportunity to present our technology here today in my hometown of Brussels. It's quite a pleasure for me. So I'll be talking in the next few slides about Dynamap, which is a precision metagenomics tool that goes beyond sequencing. And beyond sequencing in this case is definitely not a sales pitch. It is really sequencing-free microbiome analysis. So we take this quite seriously, and I'll explain in a second how exactly we do microbiome analysis without relying on sequencing the actual sample. So first of all, why was this be needed in the first place? Well, we think that, and we've heard it today before, that strain specificity is really becoming more and more important, and is really where all of the function of a bacterium is happening. So strain specificity is needed in the field of microbiome analysis, yet it's still very hard to achieve that. Of course, with 16S amplicon-based sequencing, it's entirely impossible to reach a strain level resolution. But even with shotgun sequencing, it can be relatively hard to reach, and it comes at a high cost. So low resolution is definitely one of the issues that the microbiome field is facing. And besides that, I'm sure everybody has faced this sensitivity issues in detecting your strain of interest, but also high noise, so low data to noise ratio, basically, low repeatability in your sequencing-based microbiome analyses. And on top of that, you're sometimes faced with complex and laborious workflows with long turnaround times. So in our approach, we follow these 5 steps. And the first step will, of course, not surprise you. In this first step, we do high molecular weight metagenomic DNA extraction. We've optimized this specifically for gut microbiome samples, but we've worked with other types of samples as well. And so what we do here is extract really long DNA molecules, 40 kilobase pairs, up to 50 kilobase pairs long DNA molecules, so extremely long metagenomic DNA. Then you would expect that we now go to a long-read sequencer and analyze the DNA that way, but that is absolutely not what we do. Instead, we apply our high-density fluorescent labeling method. And the way this works is we have enzymes, methyltransferases in fact, that target a specific 4-nucleotide word on the DNA. A word that we very commonly use is TCGA. So every occurrence of the 4-nucleotide sequence TCGA will receive one fluorescent molecule from our labeling mechanism on this. And so you can now imagine that the DNA, the very long DNA molecules that we have extracted from the sample, they have been labeled very specifically with one fluorescent molecule at every occurrence of TCGA. So now a fluorescence pattern is formed on the DNA molecules. And it's this fluorescent pattern that allows us to recognize the bacteria, the strain of interest in this case. What's important to realize here is that the TCGA sequence has no meaning, and it's just short enough so that it occurs on every bacterial genome with a decent frequency. On a random sequence, as you know, it will occur once every 256 base pairs. So on 50 kilobase pair long molecules, you'll have around 200 of those. This whole labeling reaction is a reaction that takes, including cleanup afterwards, around 60 minutes. So that's a pretty quick reaction. You'll notice there's no PCR reaction involved at this point, and there's also no library preparation involved. Instead, after we have labeled the DNA, so we've now created these fluorescence patterns along the DNA that reveal something about the underlying sequence, what we do next is we do molecular combing of the DNA. I'll show you a movie of how this actually happens in practice, but for now suffice it to say that we linearize those labeled DNA molecules on a flat surface. Once this is done, we will have prepared around 10 gigabase pairs worth of labeled DNA ready to be imaged. And the imaging is done in the next step, of course, which takes about 20 minutes for one gut microbiome sample. So collecting the data equivalent to 10 gigabase pairs worth of DNA. So this is an automated microscope taking pictures of the labeled fluorescence molecules. I'll show you a picture of that later, and automatically scanning this and moving on to the next sample. And in the final step, we take all of these fluorescent patterns and we compare the measured ones against a pre-computed reference database. And so that means that we will take every molecule and align it against a fluorescence pattern that has been computed from a known reference sequence. So imagine you have E. coli with a known reference sequence. You download your reference sequence, you convert it into the optical map. We have the code for that, of course. Then we will simply compare every measured molecule against every possible reference profile that we computed from a reference sequence in this algorithm. So that allows us to build a reference database of up to 100,000 reference genomes. So you can basically multiplex the detection of all of these just like you would for shotgun sequencing, of course. This whole classification algorithm is also very time effective. It only lasts about half an hour to do that. So I promised you a movie about how the optical mapping actually works. So you can see on the left an animation of the process, and on the right an actual movie. So on the left you see this droplet with fluorescently labeled DNA that is being rolled over a strongly hydrophobic surface. And what happens is that the DNA molecules, they will start to tether when they're in contact with the surface. They will start to tether with their endpoints to the surface. But then, of course, at some point, the meniscus recedes beyond the tethering point, and the molecule comes out of solution and is being pulled on by the droplets, and it actually becomes 1.7 times longer in this process. And on the right, you can see this actually happening under a microscope. So you see towards the bottom, you see the rolling droplet itself, and at the interface of the droplet, you see those lines coming out. Here, for example, there, those are the individual DNA molecules coming out of the solution and being stretched in this method. This is what the fluorescence patterns look like under the microscope. So this is really our raw signal. So you see the vertical lines that are highlighted here. These are individual double-stranded DNA molecules that carry the fluorescent labeling pattern. So this pattern, and I'm zooming in on one here, reveals something about the underlying surface, underlying sequence, of course. It doesn't allow you to read the sequence, but it contains sufficient information to determine what strain of organism this is. In this case, it was a Saccharomyces cerevisiae of 54 kilobase pairs long fragment. So this pattern is our raw read. Now, this is where I like to highlight something, which is that these DNA molecules that you quite literally see, it's an optical microscope, these are the very DNA molecules that were in the bacteria, or yeast in this case, of the original sample. And that is, of course, quite unique, because with sequencing, you always either do a PCR reaction for 16S amplicon sequencing, which introduces many kinds of bias, as we all know. And secondly, if you would use shotgun sequencing, you would still synthesize new DNA in order to read it out. It's still sequencing by synthesis, which are all causing different forms of bias that we don't have. In this method, it's a very linear, direct method. You literally see the DNA that was in the original sample. And on top of that, it's a very high throughput method because we scan hundreds of thousands of molecules in less than 20 minutes, which means gigabase pairs, the equivalent depth of shotgun sequencing, of deep shotgun sequencing. Now, I promised in the beginning that we can achieve strain-level resolution with this method, and this may come as a surprise because by now it will be clear that we do not have single nucleotide resolution. With this method, you cannot see single nucleotide differences between two genomes. However, we look at very long fragments. We look at 50 kilobase per long fragments, and that means that we start to become very sensitive to another phenomenon that happens and that causes strain differentiation, which is, of course, structural variation. And especially in bacteria, this is a very common mechanism. I'm showing here some example of Yersinia pestis, different strains. The blocks that you see here, the colored blocks, indicate regions of the genome that are identical between the different strains, but have been reorganized in the structure of the genome. So they can have extremely high ANI, above 99.5 or more percent, very high average nucleotide identity, and yet represent themselves very differently at the scale that we're looking at of 50 kilobase pairs. And that's where we are with our method, with Dynamap, extremely sensitive. So that I will show some data, of course, later. That is really the mechanism that allows us to do microbiome profiling at the strain level. So for some validation data now, first of all, we were wondering, well, does it work at all? We were analyzing mock microbiomes, of course. This is a Zymo mock microbiome that contains 3 easy-to-lyse gram-negative bacteria and 5 tough-to-lyse gram-positive bacteria. And we first tested, can we detect all of them with our extraction protocol for the high molecular weight, with our entire method. And of course, yes, we can. We've got a 100% recall rate on that. There's no significant gram-positive versus gram-negative bias, which is important. And then secondly, we were wondering, what about the limit of detection? How low can you go in relative abundance and still detect it? So we spiked into the mock microbiome mix, we spiked Vibrio HRV at 1%, 0.1%, and 0.01%. And then we compare this against shotgun sequencing in these stacked bar plots. So what we see is that the Dynamap results for the 1%, the 0.1% show a very easy detection of this, but at the 0.01% we're below the limit of detection. With a bit more details can be found on this in our recent preprint here on bioRxiv. If you're interested, I can send you the link. We have a booth just here down the center of the auditorium, and you can come talk to us about that. But here, what we found is that the limit of detection is around 0.05%. Now, for the strain level resolution, what we did was we took the species that were in the mock microbiome mix earlier. In this case, I'm focusing on Enterococcus faecalis, and we downloaded all of the reference sequences that we could find with complete assembly from NCBI. As a test case, we of course know what the ground truth strain is, and now we test it with a huge reference database of thousands of potential alternative strains. We tested, do we identify the correct one? And so the most abundant one that we identified was indeed the ground truth strain. So that's good news, of course, but we also see 2 false positive genomes being identified here. Now, the important question is, what average nucleotide identity do they have with the ground truth? And what we find, that's what this tree graph shows, is that their ANI is higher than 99.9%. So that, of course, brings us really into the strain resolution case. What's more, even the low abundant false positives have an ANI that is higher than 99.5%. So we can confidently say here that strains with an ANI higher than 99.5% can be differentiated with Dynamap. And we tested this on many other species as well. And the same holds true every time. It's between 99.5 and 99.9% average nucleotide identity. We can nicely differentiate those strains. Even though we don't have single nucleotide resolution, it's all thanks to, of course, the structural variation between these strains. You might also be interested, but how does it compare on a gut microbiome sample with shotgun sequencing? And this is one experiment where we looked into that. We have here one fecal sample analyzed 5 times with our method, Dynamap, which is a version of optical mapping. And then we also analyzed the same DNA with a whole genome shotgun sequencing. So you can see there's relative abundance that is plotted here at the species level. And the 0 line, this is the case where if you have a perfect match between Dynamap and shotgun sequencing, then you would expect a mirror image between the two. So you see for many species that is the case, and there's only minor differences really between some of the species detected with both methods at the species level. At the phylum level, the correspondence is very nice, of course, between the 2 methods. To give you an idea, quantitatively, the log-log correlation at the species level for relative abundance is between 70 and 80% between shotgun sequencing and Dynamap, so very high identity between the 2. So to summarize these results so far, what we've developed with Dynamap, with this precision metagenomics methods that goes beyond sequencing, is a method that has much less sources of bias than the standard sequencing methods, because there's no library prep, there's no PCR amplification, and there's no sequencing by synthesis. So we skip all of those potential sources of bias. There's a high specificity that we see because we can identify strains, even though their ANI is higher than 99.5%, and that is thanks to these very long DNA fragments and the high density labeling that we apply. And finally, the sensitivity can go a little bit better than 0.1% relative abundance. You can use this technology today we offer this as a service from our central lab here in Belgium, actually not so far from here in Leuven, where we analyze your microbiome samples with a 5 business days turnaround time. So if you're interested in trying this out, if you have big studies lined up, please come and talk to us. We're just down the hallway here to the left. for information on this. And then even more excitingly, this year, soon in a few months, we will be launching this technology as a benchtop device. This device fully automates the workflow, takes in your extracted DNA, extracted with our extraction kit, takes in the consumables that do the labeling reaction and the molecular combing. So the whole device automates all of this on 8 samples in parallel and produces results, gut microbiome profiling results, strain identification results in 6 hours. So with that, thank you for your attention, and I'm very happy to take any questions you might have.
Thank you so much. Thanks, Arno, for the time. We'll keep the questions for later.